AI is a powerful tool.
This summit can help ensure that artificial intelligence charts a course that benefits humanity and bolsters our shared values.
Welcome to AI for Good, the leading action-oriented, global and inclusive United Nations platform on AI. Organized by ITU, in partnership with 40 UN sister organizations, and co-convened with Switzerland. The goal of AI for Good is to identify practical applications of AI to advance the United Nations Sustainable Development Goals and scale those solutions for global impact. In today's session, we're counting on you to use the live video wall feature to ask questions and post comments to help create an engaging discussion. We encourage you to stay until the end to chat, connect, ask questions, and network with our distinguished panelists and world-class AI experts in the neural network. It is now time to kick off the session and welcome our first speaker. The floor is yours.
Hi, everyone, and thank you so much for joining us this morning. My name is Harrison Zhang, and I'm a researcher at Harvard Medical School and going to be the moderator for today's event. So we are really excited today to be joined by Dr. James Zhou from Stanford University. James is an assistant professor of biomedical data science, computer science, and electrical engineering at Stanford. James develops machine learning methods for biology and medicine, and specifically, he works on both improving the foundations of machine learning by making models more trustworthy and reliable, as well as in-depth scientific and clinical applications. James's work has been supported by the Sloan Fellowship and NSF Career Award, 2 Chan Zuckerberg Investigator Awards, a Top 10 Clinical Achievement Award, several Best Paper Awards, and faculty awards from Google, Amazon, Tencent, generative AI in biology and medicine. And without further ado, James, the floor is all yours.
Great, thank you very much, Harrison and the organizers for the introduction, for the invitation. So I'll go ahead and share my screen. Great. Yeah, so really excited to be here and then to discuss with all of you, I think, some quite exciting recent advances in generative AI and how we can use generative AI to advance biomedicine and healthcare. So as Harrison mentioned, my name is James Zhou. I'm a professor at Stanford University. So there has been, I think, quite tremendous advances in generative AI over the last few months and a year or so. So generative AI is a relatively new class of AI techniques where the output of the model can be very complex and flexible. So it could be outputting text or images, or as we'll see, molecules or even more complex objects. So here's an example of this kind of text-to-image generative AI model, right, where you can type in a piece of text and you have models that are running diffusion that can generate images based on your text, right? So for example, if you put in text Mario on trial for war crime, this would be an example image produced by these generative AI algorithms. And these kinds of images can then be very useful for, for artistic applications and also for education and other applications. Another example of generative AI that many of you have heard about is GPT, ChatGPT, GPT-4 type models. right? So these models have been tremendously powerful for answering questions, right? And almost similar as the purpose of a search engine. So as an example of this, so we've recently investigated how well these GPT-4 models can actually do in answering medical questions. Right, so the kinds of questions we asked came from the US medical license exam, which is a standard exam that doctors have to take here in the US to get their board certifications. And here's like an example of these questions from the exam. Right? In this case, it's asking about how would someone estimate the risk reduction from two different procedures, right, for treating infections. And this is the output of the AI model, which in this case, it gets the right answer. But it's also quite interesting to see how GPT thinks through the problem, right? So in this case, GPT would say, okay, well, let's look at Wikipedia articles to first find out what is the definition of risk reduction. And once it gets that definition, then it applies that formula to the specific numbers from the question to derive the correct answer. Right, so actually going through this thought process from the AI model itself can be quite informative and educational for potential for students. Now, these are, I think, really exciting applications. But what we want to do, especially in science, is to go beyond this and see, okay, now beyond answering questions, right? So are there sort of deeper applications where these generative AI models can help us to really make fundamental advances? Right. So a lot of the research that we're doing in my group here at Stanford is on how to develop new generative AI approaches, and then also to make these algorithms really helpful for biomedical scientists. And in our research, we've identified sort of three broad areas where we believe that generative AI can be quite transformative for researchers. Right. So the first area is around using these generative AI approaches to really expand and augment the creativity of biomedical researchers. The second direction is on using these models to help us to balance complex trade-offs, because in many of these scientific applications and biomedical applications, you have to balance different trade-offs like the safety of the patients versus the efficacy of the drugs, right? So you want to, these models can be very helpful in striking good balance. The third application I'll talk about is on how to use these generative AI models to help us to turn relatively inexpensive data that are widely available into much more expensive and rich data that are hard to collect. And I will illustrate each of these three directions with a concrete application, with a little research vignette. Right, so for the first direction, I'll talk about how we can actually use these generative AI to help us to develop and to validate experimentally some of the novel antibiotics for very dangerous pathogens. And then the second part about balancing trade-offs, I'll illustrate this with the example of how we can use these generative AI approaches to help us to design clinical trials, which is really one of the most important steps in the entire medical translation pipeline. And then the third part, I'll illustrate this by talking about how we can use generative AI to create what we're calling in silico spatial omics. Right? So spatial omics is a very important type of data that are hard to collect, but extremely rich and powerful. And we'll use these AI approaches to help us to computationally generate these in silico omics. So we'll start with then by looking at the antibiotics. So the history of antibiotics is actually quite interesting, right? So many of people know that it's first discovered by Alexander Fleming, who discovered penicillin in 1928. And in the first few decades after the discovery of penicillin, there's actually quite a large number of new antibiotics that were discovered, right? There's sort of like a gold rush of different molecules. And what's really important is that basically for the last 60 years, there were relatively few really novel antibiotics that people were discovering, right? And this is a huge problem because if the antibiotics that we have are not very different from each other, then it's easier for bacteria to develop resistance across the spectrum of antibiotics, right? And bacteria that have resistance to antibiotics is now a huge public health challenge, right? So currently, over 1 million people die every year because of bacteria that are resistant to existing antibiotics. And that number is estimated to really explode over the next few years, so that over 10 million people might die from antibiotic-resistant bacteria. And the bottom here is actually a plot that illustrates some of this. It's the top is a timeline. So above the timeline shows us the different antibiotics and the year where it's introduced, right? And the bottom of the timeline shows the year where we first observe resistance, bacteria that have resistance to an antibiotic. And what's really quite scary is that you see this arms race between new drugs and bacteria that develop resistance. And in many cases, right, so the bacteria only takes a few years before developing resistance to the new drugs. And we're really losing this arms race, right? And as a result, we have these bacteria now, for example, this one called Acinetobacter baumannii, which is considered by many to be the perfect predator because it's resistant to many of the existing antibiotics that we have on the market. And our goal here then using AI is to be able to develop the first novel molecule antibiotic that's effective in killing Acinetobacter that's developed by an AI algorithm. Right, so I'll show you how we do that. So our goal here is to use AI to help us to develop new molecules, molecules that have never before seen, and molecules that are also effective in killing these bacteria that have resistance to the existing drugs that people have. This work is led by Kyle Swanson, who is really a fantastic PhD student in my group here at Stanford. And I'll describe a bit more of how Kyle solved this problem. So the key challenge with using AI, right, to generate new molecules and new drugs is that these molecules that are generated by AI are often very difficult to synthesize, difficult to produce, right? And this is a big problem because if you can't really produce these molecules, then you don't really have the drugs in practice, then it doesn't really help anyone. So the approach that we took to address this challenge is to not ask the AI just to simply produce new molecules, but also ask the AI to explicitly produce the recipe for creating that molecule, right? So the AI is basically going to give us the step-by-step recipe, the synthesis pathway for efficiently generating each of the new molecules that it recommends. So here's how the recipes would look like, right? So the recipes would start with these different fragments of molecules that we know are available and easy to work with, right? And we can put these different fragments together with chemical reactions, right? So these are also known chemical reactions, right? So each reaction then could apply to a subset of molecules and generate more complex output. Right? So this is a huge search space because there could be over 100,000 of these fragments, and there are hundreds of these reactions, and you can use them in different combinations. So the search space of potential molecules is really astronomical. And our goal here then is to basically use the generative AI, right, to help us to explore the space of all these potential molecules, and also to come up with the different reactions and fragments for producing each of the molecules. Okay, so here's an illustration of how this works. So we use a type of machine learning algorithm. It's called multicolored tree search. It's actually quite similar to the algorithms behind AlphaGo or AI systems that are used to play Go and play chess. Okay, so here's how it works. So it's basically going to search through this tree where the leaves, the nodes of the tree and the leaves are the individual molecules that we're trying to, molecules of interest. Right, so the early initial levels, right, the level 1 of the search tree corresponds to these simple fragments. Right? And then level 2 then corresponds to, you can take combinations of these fragments and put them together with reactions into more complicated molecules. Right? So basically, as you go further down into the tree, we're creating more and more complex molecules. Right? And the AI algorithm, the multicolored tree search, is going to search through the particular path of these this molecule tree to explore unseen molecules that we have not observed before. And what's doing this is guided by what we call property predictor, which is basically another machine learning algorithm that tries to estimate how good is potentially a new molecule that the AI algorithm is synthesizing. Right? So this is all done in silico in computation. So the property predictor is another neural network, right? So basically it takes each of the proposed molecule, represents it as a graph, and then runs a graph neural network on top of this molecule to estimate like how effective this molecule potentially for killing the bacteria of interest. And then it generates a score, right? The larger the score means that this molecule is more likely to be effective. And that score then is sort of back propagated into the nodes of the tree to update the tree. Right, so the score then basically helps to guide the multicolored tree search to explore parts of the molecule space that are novel, but also are likely to be effective. And this is quite important because the space of potential molecules, as we saw, is really astronomically large. So this property predictor basically helps to guide the AI to explore the most promising part of the space. And when the AI algorithm is exploring this part of the space, it's trying to balance these different competing objectives. Right, so first, it wants to develop molecules that are likely to be effective. Right, so it wants to have high efficacy. As we said, we also want to have molecules that are structurally novel, which means that it's very different from any of the existing molecules that people have used before. Right? In addition, we like these molecules to be safe for humans, right? So they have less toxicity and low adverse events. And importantly, we want these molecules to be very easy to synthesize. Right? So by balancing these different objectives, then the AI is going to explore and go down this path of the tree and generate recipes, where each recipe then corresponds to an explicit path that reaches a particular leaf. And the leaf one corresponds to the molecules that we want to design. So we trained and developed this AI model with this multicolored tree search, right? And everything down here is sort of down on the computer, right? And then the fun part starts, right? So now we can actually take these molecules that are created by the AI and actually make them and test them in the lab. So to do this, we actually collaborate with a company based in Ukraine, right? It's called Enamine. They're based in Kiev. So the AI algorithm generated 70 recipes. Each recipe corresponds to a step-by-step procedure for creating an entirely new antibiotic molecule. So we sent these 70 recipes to Enamine. And you can imagine that over the past year, they have a lot of things going on in Ukraine, in addition to making new drugs. But amazingly, Enamine was still able to synthesize 58 out of the 70 recipes, right? So there's a success rate above 80%, which is extremely high. And one reason why this has been very successful is because the recipes created by the AI, right, are very easy to synthesize, and they're very robust. And then the next step is that we take these 58 molecules. So each of this is sort of a new novel molecule that have never been seen before. And then we test them across different bacteria to see how effective is this molecule in killing the bacteria. And to do this, we actually picked a set of bacteria that are considered to be escape pathogens. So escape pathogens is a classification by the World Health Organization to be the most dangerous bacteria that have developed resistance to the existing antibiotics or the existing drugs that we have. So there are 6 escape pathogens classified by the WHO. We tested our drugs against 4 out of the 6 escape pathogens already, and we're testing it on the other 2 going forward. And what's really exciting is that we have 6 novel molecules here that have shown to be experimentally effective in killing these 4 out of the 6 escape pathogens. And this includes this Acinetobacter baumannii, the perfect predator. It also includes other pathogens that are responsible for killing many people every year. So here are the 6 molecules that are developed by the AI algorithm. And these plots here, which we don't need to go into detail, are just showing the experiments validating the effectiveness of each of these molecules in killing a range of different bacteria. And what's also important is that each of these 6 molecules, they are effective when they're used at low dosage. And this is important in reducing the potential toxicity for humans. But we need to do still further animal model testing to really ensure that these molecules have low side effects. But we're really excited about this work, and it's an example of one of the first examples of really novel molecules that are created by the AI algorithm and then are shown to be effective as antibiotics across these dangerous pathogens. So that's the first direction. And then I want to show you about the second part, which is how we can use AI to balance complex trade-offs. And we want to go illustrate this by talking about how to use AI to develop clinical trials, to make these clinical trials more efficient and also more inclusive, more diverse. And this work is led by Rishan Liu, who's another awesome PhD student in my group at Stanford. And to describe this paper we published in Nature, where we proposed this AI platform called Trial Pathfinder for helping to design clinical trials. And we're excited to say that Trial Pathfinder is now actually being used by large pharma companies, including Roche and Generative, to help them to inform the design of new cancer trials. It's also recognized by the pharma industry as the top data science innovation last year. So clinical trial is really one of the most important steps, is the bottleneck of the entire medical translation pipeline. Right, so we can start with these hundreds of new drugs, maybe new antibiotics, but only one of them would actually make it to a patient because they have to go through these different stages of very strict clinical trials. And each trial itself can cost on the orders of tens, sometimes hundreds of millions of dollars, which means that the cost of actually bringing a new drug to patients, considering for these many trials that are needed, is on average several billions of dollars. Right? So anything that we can do to make clinical trials more efficient and more effective will be broadly beneficial to the entire medical translation pipeline. So one of the reasons why these clinical trials are very expensive and difficult to run is that they're highly selective, right? So maybe you have a trial that's looking at a particular disease, and among all the patients with that disease, only a subset of the patients are actually eligible to participate in that trial, right? So what the trial is doing is that actually lists out different criteria, different constraints to filter out patients explicitly. So some of the patients are filtered out because they have comorbidities. Other patients are filtered out based on other previous conditions or based on if their lab values are outside of some range. And what the trials are doing when they're trying to filter out the patients is that they're basically trying to look for what are so-called Olympic athletes among the patients. Right? So they want to look for sort of the healthiest among the patients where the drugs can be effective. And people are recognizing now that most of the clinical trials are just overly restrictive. They're filtering out too many patients. And this is a huge challenge to the drug developers in that a large number of trials have a hard time to recruit enough patients. So 40% of cancer trials fail to recruit enough patients. And it's also really harmful for the patients because this means that if the trials are really selecting for these Olympic athletes of the patients, then how well the drugs work during the clinical trials, right, on these Olympic athletes of the patients may not reflect how well the drugs work on average patients like us. Right? So there's a huge distribution shift. And moreover, these clinical trials are often not very diverse, right? They're tested on very small slice of the patients, which do not really reflect the general patient population. So, how do people currently design these clinical trial conditions then? Right. So here's just a few concrete example of this. I took a screenshot from our colleague at Roche of a trial that is being designed, being developed. Right. So the first thing that people do will be, so each of these rows here corresponds to a different criteria called eligibility criteria for filtering out patients. So the details here are not very important, right? But the first one basically says that there's a particular lab value called neutrophil count, right? And if the neutrophil count is too large, then those patients might be filtered, right? And you can actually see an interesting discussion in the comment section of the Google Doc, where some clinicians actually asked, can we relax that threshold to enroll more patients? And another clinician will actually say that, well, we don't really have evidence to change that threshold. So then let's just use what was done before to be consistent. And as you can see from this discussion, because of the lack of data-driven approaches to help these clinicians to design the trials, oftentimes people just fall back on the trials that was done before or the more conservative criteria. As a consequence, right, these trials are very restrictive, and there's a mandate from the US National Cancer Institute and from other organizations that these clinical trials really need to be simplified and enroll much more diverse, more patients. But how to do this is the open challenge, and this is where we think that generative AI can be very helpful. So our idea is to basically use generative AI, combine that with large-scale medical records to help us to emulate clinical trials in silico. And we're going to use these in silico trials to help us to design new trials, to decide what patients to enroll in new trials. And to do this, we partner with the Flateral Network, which have medical records from over 2 million patients from hundreds of cancer hospitals and clinics across the US. So here's an illustration of how the generative AI approaches work. Right, so the algorithm is basically going to propose a set of eligibility rules, similar to what clinicians are currently doing. So each of these rules then corresponds to a specific constraint, right, based on lab values, different biomarkers, for deciding what patients to enroll in that trial. For a given set of rules, right, so then it selects for a subset of patients. Then the algorithm goes into the medical records, takes those rules and apply that to hundreds of the hospitals, and to select which patients from these hospitals are eligible under the rules, and also this selects a set of control patients who are taking similar drugs, right? And then we can statistically compare these two in silico distributions of patients to remove confounders, and then to estimate like what's the outcome of these patients who are taking the treatment, whether they're safe or whether they have toxicity events, and also quantify how easy is it to implement this rule and how many eligible patients there are in the population. Because this is all done in silico, right, so the algorithm can very easily propose a new set of rules, right? It can modify these different thresholds or remove some of these rules. Once doing this, right, now the rules become more relaxed. When it goes back to the medical records, we can identify more patients that satisfies these constraints, right? And then we can evaluate when we add in more patients, how does that affect the outcomes, the survival and the efficacy or the toxicity of these new patients, right? So by doing these comparisons in silico, you can get a data-driven estimate of what is the impact of changing and relaxing certain eligibility rules to enroll more patients. And as you can imagine here that there are many, many millions and millions of combinations of eligibility rules, right? So this is where the AI algorithm has to, similar to what we did with the antibiotic molecule search, here the algorithm has to search through these different potential combinations of rules to identify what set of rules are good for balancing these different trade-offs. So this is in a nutshell our trial Pathfinder algorithm and approach, right? Basically it uses these generative AI plus the medical records to design a set of eligibility criteria that balances these complex trade-offs. And it's doing these millions of in silico trials in parallel, right? Each in silico trial then is using a different set of combinations of rules and different thresholds. And for each of these in silico trials, it uses the real world data to evaluate the outcome, the impact of those eligibility rules in terms of the outcomes of the patients. And in the end, the algorithm summarizes the information from these millions of in silico trials into one set of data-driven recommendations for the patients on how to for designing these eligibilities of the clinical trial. Right. So here's an example kind of output. As for each of the particular trial, the trial Pathfinder AI algorithm then will recommend what set of combinations of eligibility rules, which are the columns here, are the most appropriate for that trial in order to balance the safety of the patients and the efficacy of the treatment. So we applied this approach to a variety of different diseases. So here I'm just showing you some example applications to lung cancer, which is the most common form of cancer. Here we applied the algorithm to 10 different clinical trials that are looking at advanced small cell lung cancer. Each row here is one of these clinical trials. So the first two columns, and we can focus on the last row here, which is the average across these 10 different trials. So the first two rows indicate if we have used actually the original designs of these trials, right, the original eligibility rules, here's how many patients that we'll be able to enroll on average. And here's what's the outcome of these patients evaluated in terms of the hazard ratio. The hazard ratio, lower is better. And then the last two rows corresponds to if we actually use the AI design, the data-driven designs of these clinical trials, we see that we're able to enroll a lot more patients. So on average, we can more than double the number of eligible patients. What's also really interesting is that the hazard ratio of these patients, even though we're enrolling more patients, the hazard of these patients is comparable, it's even smaller than before, which suggests that many of these new patients that will be enrolled under the AI criteria would benefit safely, right, from these treatments. They benefit as much from these drugs and treatments as compared to the Olympic athletes of the patients. And this is critical for inclusivity, because by using this data-driven recommendations proposed by the AI algorithm, we show that we can enroll more women, a lot more minorities, and also more older patients. So many of these groups were systematically excluded from participating in clinical trials before, right? Often because maybe they don't fall within some narrow range of lab values. But the AI algorithm has learned that we can actually relax those lab values safely and to enroll a lot more of these patients, which will make the clinical trials much more diverse, and also makes the results of these clinical trials more representative of how well the drugs will work on real patients. So this approach, this trial Pathfinder approach, right, I think demonstrates that we could use this generative AI and data-driven designs to make clinical trials more inclusive and more diverse. I think this just really benefits the patient. It also benefits the drug developers because it makes it then easier for them to enroll and to recruit patients. And we've applied this to several different cancer types, and we're also have validated this approach using data from randomized clinical trials and different cohorts. Right. So now we're excited that it's actually being used by the large pharma companies for guiding the designs of their new trials, and we have received several awards. Okay, so the last vignette I want to describe is to illustrate, how we can use generative AI to help us to turn relatively cheap, inexpensive data into really powerful, much richer data, right? And this is what illustrate this was this idea of in silico spatial omics. So spatial omics is a really exciting new kind of technology that people have been developing for the last couple of years, right? So you can generate really beautiful images like the one shown here in the background, where you can actually see the individual cells, right? These are the individual nuclei here, and the different colors indicate the different proteins, right? In this case, right, so spatial proteomics, so we can measure up to 100 different protein expressions in parallel at single cell resolution on these tissues. So it's a really powerful way to study cell-cell interactions and also study different disease microenvironments. So the big challenge is that these kinds of spatial omics data is extremely difficult to generate, and it's very expensive, right? It costs several thousands of dollars to produce each of these images. So then we asked, okay, can we actually use AI to help us to generate these spatial omics data? And with the idea that the AI generated spatial omics will provide similar insights, have similar utility as the really expensive experimental spatial omics data. So the first proof of concept that we propose for doing this is to generate these spatial transcriptomics directly from what are called H&E images. So H&E images are these very routine histology images that are collected in pathology departments, right? So it's very easy to collect. It costs a few dollars. And these are very routinely collected from patient biopsies. And now we ask, okay, can we actually take these H&E images and use AI algorithms to essentially to up-level these images into these more spatial gene expression patterns, right? So basically the algorithm then is performing like an image-to-image translation. It takes this H&E image as the input and then computationally generates hundreds of new images. Each of the new image then corresponds to the spatially resolved gene expression pattern of a different gene of interest. Right, so in this case, right, so it takes this image and predicts like where is this gene FASN highly expressed, that's the yellow regions, and where it's lowly expressed, that's the blue regions. And FASN is one of the key breast cancer markers. For a different gene, right, it produces a different output image describing its different expression patterns. Right, so we demonstrate that this algorithm, we are able to in silico generate these spatial gene expression patterns directly from the H&E image for several hundred genes. And these generated images are very accurate, so we can compare with the experimental measurements, which are the more things that are more difficult, expensive to collect, right, using the platform called Visium. And compared to the experimental measurements, we can show that the computationally generated spatial transcriptomic images matches up very well with the experimental measurements for these hundreds of genes. And the algorithm is doing this essentially by zooming into the histology image and by analyzing the morphologies of the individual cells, their nuclei, and their neighborhoods, it extracts the features about the morphologies and uses those features and then to generate the gene expression patterns. And we think this is a really powerful approach for making these kinds of spatial omics data much more broadly available. Right, so the idea is that that these H&E histology images are very routinely collected, right? So we can basically apply the algorithm now as sort of like a virtual filter on top of the histology image, and this filter now is going to create like these different gene expression patterns for different genes of interest, which then the researchers or the clinician can use to help to inform their diagnosis or to inform their research. Okay. So this is an example of how we can use AI to take data that are relatively easy to collect, very inexpensive data, and then computationally turn them into data that are much more expensive and much richer. Right. So we've also demonstrated in previous research how we can use these AI to help us to improve the quality of the data. Right. So one challenge with the experiments with these spatial omics is the spatial resolution. Right. So here's the standard setups, right, with the experimentals using this company from 10x platform called Visium from 10x Genomics. And how this works is that it's basically generating like fairly pixelated samples of the spatial gene expression patterns, because the probes for the experiments are fairly coarse probes, right? So each of these dots is one probe, and it covers like a region of a few dozen cells. And it's basically taking like an average gene expression from this neighborhood of 20, sometimes 50 cells. By leveraging the AI algorithms, we show that we can actually take these coarse measurements and then computationally perform super resolution, right? So that the output of the model plus on top of the coarse measurements is of much higher spatial resolution at sub-single cell resolution. And then we validated that when you compare the output from the AI model, right, this super resolved output from the AI model with more expensive and targeted experiments looking at these high resolution gene expressions, they match up really well. Right, so this demonstrates that we can actually then use AI to improve our data from low resolution to high resolution to derive the more fine-grained biological insights. Right, so these are examples, I think, to illustrate what we see are some really exciting applications of how we can use generative AI to up-level our data to turn the more readily available noisy data into high-quality richer data. So far we've illustrated to these three, I think, quite exciting directions of how generative AI can really improve biomedical research. I think there are also some important limitations and open challenges that we want to be aware of. Right. So one open challenge that we've identified recently is that these generative AI algorithms can often amplify human stereotypes. So here's the study that we did to illustrate this, right? So I mentioned in the very beginning of these text-to-image models where you can type in a piece of text and generative AI would then produce new images based on your text descriptions. So here we asked a very popular example is called Stable Diffusion to generate images based on these very simple texts, right? We can tell it, okay, produce it for me an image of a software developer or an image of a poor person. And here are basically the representative images that are produced by the generative AI models based on these simple text descriptions. And you can see that there's algorithms are actually really contains quite a lot of stereotypes, right, for each of these occupations, for each of these descriptions, right? It has very stereotypic views of what a poor person would look like, also what a terrorist would look like. And that's also very stereotypic views of what a software developer look like, right? So for example, there are no women generated among these images of software developers. And what's really important to note here is that it's not just the algorithm is reflecting biases in the data sets, right, that's used to train the models, but the algorithm is actually actively amplifying the existing biases and stereotypes in society and in the data sets. And we can measure this amplification effect. Right. So, for example, for each of these occupations, we know in the data set and also in the census, right, what fraction of the population, in this case in the US, who have that occupation was self-identify as male or female, right? So for example, for software developers, right, so about 20% of the software developers actually identify as female. So that's the red, that's the vertical bars. And the algorithm, however, would produce essentially no software developers in its output for based on the prompt of software developers. Right. So this is actually an effect where the model is not simply capturing the existing imbalance in the data, but it's actually actively amplifying these imbalances to the extreme. And similarly for these other occupations. And we think this can be potentially very dangerous if, for example, these generative AI models are producing these data, which are then used in education materials or used as additional data to train other AI models, right? And then these kind of bias amplifications can propagate throughout the society. So this is an important open challenge that we and our colleague are trying to address in ongoing research. So just to summarize, right? So here we think that these generative AI, first, I think they can be really a powerful approach, right, for biomedical applications, right? And we saw that how they can be used to augment the creativity of researchers to design new antibiotics, how they can be used to balance these complex trade-offs of patient safety and inclusivity to help design new clinical trials, and also how to up-level our data to turn relatively inexpensive data, right, into these very expensive, very powerful in silico spatial omics data sets. And we've also discussed like how these models can have some of the ongoing challenges with generative AI, especially as it relates to bias amplifications, which is an active area of research that we're working on. So I want to acknowledge again the really amazing students in my group that led these works, especially Tayo, who led the antibiotic work, and Rishan, who led the clinical trial design work. And then here are again the references for the papers that I mentioned. And I'll happy to stop here and to have a further discussion and questions.
All right, so thanks again, James, for that extremely exciting and invigorating talk. The work that you guys have been doing is, of course, really inspiring, and we're all really excited to hear about it. So we do have a few questions from some of the audience members. The first one that I'll kind of pose to you is from Guy, and he's basically saying that many imaging modalities obtained from healthcare, such as PET scans and MRI scans, are really expensive, and they're often obtained later in the diagnosis or prognosis journey. And so I guess he's asking, do you think that there's going to be a solution with a generative AI model to help mimic these images, or to help, make them more useful for research or real-time prognosis or diagnosis?
Yeah, thank you. That's a really good question. I think also aligns quite well with like the third direction I mentioned of how we can use these AI to help us to up-level our data. So I think a lot of there is some very exciting recent research on how to use AI, especially generative AI, for medical images. Right. So for example, as we saw here, like we can take medical images like the histopathology images that are relatively easy to collect, and then use the generative AI to turn those into much more expensive data modalities. And I think people are also exploring similar ideas with X-ray scans or MRI and echo to see, okay, so can we actually use simpler, cheaper modalities, right? And then plus AI to help us to create these more expensive modalities, with the goal of making the data cheaper and also faster to produce. So that's one thread of research. The other thread of research is to say, okay, so if we have this data modalities like the PET scans, the MRI scans, or the echoes, right? Either the real ones that we collected from patients or the synthetic ones generated from the AI, how do we then extract more value or more insight from those data? Right. So that's also a very active area of research. For example, some of the work that we've been doing is on how we can take echocardiograms of looking at heartbeats, and then be able to extract a lot of other and essentially predict a lot of other lab values, right, from these echoes, and also to diagnose a lot of other diseases from these ultrasound videos. Right. So I think in both directions, there are a lot of very fruitful applications of AI.
Awesome. The next question that we have is from Tyrone. And I think this is with regards to your work on designing novel antibiotics using generative AI. And I think he's probably referring to that score that you guys use to kind of guide the molecule development. And Tyrone's just asking if that algorithm is in any way associated or similar to the one used for AlphaFold.
Okay, thank you. Yeah, thanks for that question. So I think there's some similarities and also some important differences between what we're doing for small molecules and what AlphaFold is doing. So AlphaFold is is looking at things at the protein space, right? So it's basically trying to model protein structures. Whereas for a lot of the drug discovery, the focus not on proteins as drugs, but more on like small molecules. So the representation that we use is to represent small molecules as these molecular graphs, right, which captures the individual atoms, whereas the protein representation often tends to be a bit more complicated in terms of these contact matrices. So these are some of the differences. Now the similarity is that in both of these cases, we have these neural network algorithms that can then take the small molecules or the proteins and try to predict some sort of properties, physical properties of that object. Right? In our case, the properties we want to predict often would be things like, how effective is this molecule for killing bacteria, or how toxic is this molecule. For proteins, oftentimes the kind of structure people want to predict would be things like what is the secondary structure, or what is the folding structure of those proteins. Right? So these are some of the key differences.
Great, and thank you so much for summarizing that. That clarified a few things on my end as well. And then we have another question from Perry Share. So, you highlighted that there is a potential for bias from the outputs from a lot of these Gen AI models. And Perry's asking, what do you think are the implications of the potential bias from the model outputs in all of the applications that you kind of discussed in the 3 vignettes?
Yeah, so that's also a good question. So this is definitely an ongoing area of research for the broader research community in how to mitigate biases in AI algorithms, especially for generative AI models. So in our own research, we have to be very careful in also evaluating what is the impact of our algorithms. Right? So for example, like in the second vignette I mentioned about using AI to design clinical trials. Right? So one of the things that we explicitly try to evaluate is when we take the AI-driven recommendations for the clinical trial design, how does it affect what patients are not recruited into these trials? And there we actually do find that when we take the AI-driven designs of the trials, we're actually able to recruit a lot more diverse patients, right? More minorities, more women, more older patients, compared to if we use the more restrictive criteria that's just that people have been using previously. So in this case, then, so in that application, it was actually beneficial, right? So the AI is able to overcome some of these existing biases, right, in the medical community, and able to help us to recruit safely more diverse patient populations.
Very interesting. And I guess just kind of a follow-up from my end, as we look forward to identifying these biases, potentially debiasing and mitigating the biases in these models, given that a lot of the Gen AI recent developments have been from the industry side, what do you kind of see the interplay between industry, academia, and federal agencies kind of looking like in order to basically try to debias or make these models more generalizable?
Yes, thank you, Harrison. I think that's a really important and also a really timely question. I think it is very exciting, the tremendous recent advances in generative AI. And I agree that most of these advances have been driven by a lot of the industry side. Right? And that's both an opportunity, but also poses some challenges for regulators and for academic researchers. Right? So for example, one thing that we do often recommend is to, we want to be able to audit the machine learning models to identify, like, does the model really work well across different scenarios, different subpopulations, to detect bias or to mitigate bias. To do this kind of audits will require often access to the models or access to the under the hood of what the model is doing. right, or access to the weights of the neural networks. For many of the recent models, it's actually very difficult to do those audits as independent third parties because the models are not entirely transparent or accessible. So that's actually one of the challenge going forward. So I think it's I think having much more transparencies about how these models are developed and also what data is being used to train and develop these models would go a long way towards improving the trustworthiness of these models. As I think right now, for example, a lot of the larger language model, GPT models, it's actually not clear what is the data that was used to train the models and what is the data that's used to sort of fine tune the models, or how human feedback is being used, right, to improve these models. Right? So without having that level of transparency about the data and the methodology, then it actually makes it very difficult for external parties and researchers and also the government to regulators to evaluate potential biases and applications of these models.
Thank you so much for providing that insight. I think it's, like you said, really interesting territory going forward, and we'll kind of see how things play out. Let me just make sure. We just got a new question. Let me just take a look at it really quickly. So Yep Huang is asking if you can share your insights in how the improving of the hazard ratio from your guys's TrialFinder paper helps to mitigate existing biases in clinical trial populations, and if the current set of criteria used by some of the trials that you compared to are either not complete or biased in their own way.
Okay, yes. So thank you for that question. So one thing that's really interesting that we find is that a lot of the current clinical trials, right? So how do they filter out patients? They're often filtering out patients based on several lab values. So these are things like hemoglobin counts, right, or neutrophil counts, or bilirubin levels. And they're basically saying that, okay, if the patient's lab values falls outside of some defined range, then those patients are not eligible to participate in the clinical trial. And this narrow range for these lab values are often come from much older studies, say sometimes from 70s and 80s. And what the AI algorithm actually learns is that we could actually, those ranges are way too narrow. We could actually broaden those range and enroll a lot more patients, especially the more diverse patients like women and more minorities, right? And it's actually safe, it's actually fine, right? So basically the previous range that were for these lab values often were too narrow, and they filter out a lot of the more diverse patients. But the AI actually learns that there's not a great reason for having such a narrow range, right? By looking at the medical records and data of these millions of patients. So that's actually a very concrete insight that's learned by the AI that's then useful for improving the diversity of these clinical trials.
Yes, very fascinating. And the last question that we'll take today, just so we can begin wrapping up, is from Isabel Gento, and she's asking, what do you think will be the impact of generative AI on the number of drugs that will be brought to market and their prices in the medium term? Should we expect a significant difference in magnitude and quality?
Yeah, so that's also a really interesting question. So with the generative AI, I think one thing that could help us is to reduce some of the costs of developing drugs, right? So as we saw with the antibiotic one example, right, so it's able to, we were able to actually discover these 6 new molecules relatively quickly, right? So it took the algorithms basically a few months to develop, to uncover these molecules, and then took a few more months to experimentally synthesize and to validate these molecules. Right? So the whole process has actually happened in less than a year, which is actually extremely fast for these drug development projects. Now, of course, there's a lot more steps to do next, right? You still have to do more extensive validation of the molecules in different animal models, so that's more expensive. And then in clinical trials, which as we saw is also more expensive, but also things like Trial Pathfinder can help us to make the clinical trials also cheaper and faster. So I do think that in the medium term, there will be a beneficial impact of these AI approaches to make this entire medical translation pipeline faster and more cost efficient.
Awesome. And I think your guys' recent work that's in review certainly attests to that prediction. So I think we're going to start wrapping up now. Those look like the end of the questions. I just wanted to thank first James again for joining us today, for sharing the really, really exciting work he's doing with generative AI and its applications in biology and medicine, as well as all of our audience members who have joined us to hear about the talk today. So I will hand it back to Jinu.
Great. Thank you very much, Harrison.
Of course.
All right, and thanks everyone for your great questions.
Thank you for participating in today's AI for Good session. We hope you've learned something new, innovative, and engaging in today's event. We now encourage you to continue the conversation on the live video wall in the neural network. Here you can ask questions, like and comment, share links, complete the poll, connect with interesting profiles, or speak one-on-one using the chat and video function. We invite you to explore the lobby, try the smart matching quiz, visit the virtual exhibits, poster boards, the eShop, and build your personalized AI for Good program. Let's shape the future of AI for Good.
AI is a powerful tool.
This summit can help ensure that artificial intelligence charts a course that benefits humanity and bolsters our shared values.
you.